discuss about gene regulation. Gene regulation is the mechanism of control of protein synthesis in the cell. Gene regulation. It is basically mechanism of control of gene expression or protein synthesis. In a cell, all the necessary proteins are not synthesized always continuously. They are synthesized only when the cell is required. Cell requires those proteins, and when the cell doesn't require, those proteins are not synthesized. It means the mechanism of transcription and translation that we have studied in gene expression is controlled in the cell. When it is necessary, transcription followed by translation takes place. When it is not necessary, this entire mechanism is switched off. This is known as gene regulation, the mechanism of control of gene expression. This idea of gene regulation was first proposed by two scientists, Jacob and Monod, in the year 1961, based on the experiments that they conducted with the bacteria E. coli. So first this mechanism was discovered in E. coli and later in other organisms, and now it is known that such a gene regulation occurs not only in prokaryotes but also in the case of eukaryotes. According to Jacob and Monod, gene regulation is brought about by a set of genes present in the genome for a given protein synthesis, and they called this system as Operon system. Operon system, because this system operates either to switch on the mechanism or switch off the mechanism. The Operon system has the following components. Number one, Operon system has the following components. Number one, structural genes. Structural genes. Structural genes are a set of genes that are together involved in the synthesis of a particular set of polypeptides or proteins, which later function as enzymes for a given mechanism, for a given biochemical mechanism. So, a set of genes lying adjacent to each other. A set of genes lying adjacent to each other responsible for transcribing a set of mRNA molecules. And remember, these mRNA molecules are going to translate into a set of proteins required for a particular biochemical mechanism. So, for a particular biochemical mechanism. Then there is an operator gene. Operator gene is a gene found adjacent to structural genes. Operator gene is a gene lying adjacent to structural genes. What is its function? The operator gene can either switch on or switch off the structural genes. When the operator gene switches on the structural genes, transcription followed by translation proceeds. When the operator gene switches off the structural gene, protein synthesis does not take place. Then promoter gene. This is a gene, a portion of the DNA in the operon, to which RNA polymerase molecules are found attached. Remember, during transcription we have discussed this. There is a promoter gene which synthesizes and allows RNA polymerase to continue transcription. So RNA polymerase molecules are found attached to promoter gene. So when we say operator gene switches on the structural genes, it means promoter gene allows RNA polymerase to reach the structural genes. When we say operator gene switches off the structural genes, that means the promoter gene is not allowing RNA polymerase to reach the structural genes. Then there is a regulator gene, another portion of the DNA molecule, regulator gene. regulator gene is involved in the synthesis of a specific protein. A specific protein called repressor. So whenever the regulator gene undergoes transcription followed by translation, it synthesizes a specific protein called repressor. This repressor, so the next component is the repressor. Repressor is a protein that can bind with the operator gene. That can bind with the operator gene. When repressor binds with the operator gene switches off the structural genes. When the repressor is not available, when it is not binding with the operator gene, the structural genes are switched on by the operator gene. In addition to this, there can be a substance called inducer. The name itself indicates that inducer is a substance, a chemical substance most often it will be the substrate on which the enzymes synthesized by structural genes are supposed to act, a substance which induces binding of repressor. It induces the binding of repressor to the operator gene. Then, in some cases, there can be a co-repressor. Co-repressor is a substance, most often it is the product. Inducer will be a substrate, co-repressor will be a product which activates the repressor. So these are the normal components of an Operon system. With the help of these components, the cell is able to exhibit gene regulation. How it happens, we shall see.